Welcome back, students. Today we are going to discuss Unit 5, and in this particular unit, we are going to discuss photosynthesis in higher plants part 2. Before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any issue, any concern, please contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss these points, means photosynthesis, photochemical biosynthetic phases, diversity in photosynthetic pathways, photosynthetic electron transport chain, what is photophosphorylation, photorespiration. So, in part 1, we have already discussed about photosynthesis. So, what is photosynthesis? It is a process by which green plants make their own food. Green plants make their own food, right? During this process, carbon dioxide from the atmosphere is taken up by the leaves through stomata and used for making carbohydrate, primarily glucose or starch. Okay, and this photosynthesis process takes place in the green plants parts of the plant, right? Mainly, so what is those green part? Mainly leaves. Okay, and within leaves, the mesophyll cells have large number of chloroplasts that are responsible for carbon dioxide fixation. Okay, and within chloroplast the membranes are the site for light reaction, and stroma are the site for dark reaction or chemisynthetic pathway or Calvin cycle, right? Photosynthesis, as you know, these are having two reaction type. First is light reaction and second one is carbon fixing reaction or dark reaction. In the light reaction, what happened? Light energy is absorbed by the pigments P700, P680. Okay, and these pigments are present in the antenna site, right? And there are two type of photosystem involved. One is known as PS1, another one is PS2, right? PS1 has 700 nanometer absorbing chlorophyll molecule, that is known as P700 as its reaction center, while in PS2 P680 reaction center absorb red light at 680 nanometer. After absorbing light, electrons are excited and transfer through PS2 to PS1 side. And finally, through Z scheme, we have discussed it in part 1. And finally, these electron accepted by the NAD and form NADH. And this is the membrane site. This is the chloroplast. These are the membranes where some kind of different kind of carrier molecules are there, which is utilized by the to transfer the electron from one carrier to another carrier molecule in the presence of light. This is the reaction center for PS2 where P680 molecule are present, and in this PS2 water is getting split into oxygen, electrons and proton molecule. Electron are passed from one carrier to another carrier molecule like plastoquinone, plastocyanin, cytochrome and so on, and pass from PS2 to PS1 site where the main molecule type is P700, and finally these electrons are accepted by NADP and converted into NADPH. Okay, and the protons are accumulated in the lumen site, and then these are further transferred from F0M1 particle, right, and convert NAD to ATP, and not NAD, sorry, ADP to ATP. And this process is going on in the presence of ATP synthase enzyme. So this transfer of electrons from one carrier molecule to from here to here, then like this is known as light reaction. Okay, this is known as light reaction in which PS2 and PS1 Photosystem 1 and photosystem 2 are actually participating. Okay, now, so this all we have already discussed in part 1. Now in today's video lecture, we are mainly focusing to discuss this chemiosmotic hypothesis. Okay, means proton gradient. Electron transfer we have already discussed in the part 1. Different type of carrier molecule are responsible to pass from one level to another level. Okay, while proton, how proton transferred and how these proton utilized by the F0 F1 particle for the synthesis of ATP. Okay, so this process during this chemiosmotic this hypothesis tells us that a proton gradient is created across the membrane of thylakoid. Okay, and the breakdown of proton gradient occur due to the movement of F0 particle part of the ATPase enzyme, which is released enough energy for the synthesis of ATP. Right? During the splitting of water, where it is associated with the PS2, just now we have seen that particular picture. Okay, so splitting of water molecule is associated with PS2, resulting the release of oxygen, proton and electrons to PS2. Right, so the chemiosmotic hypothesis explain ATP synthesis in chloroplast. ATP synthesis in chloroplast. Okay, so ATP means adenosine triphosphate is produced by utilizing a proton gradient across the thylakoid membrane. These are the thylakoids. Okay, this is the outer covering of chloroplast, this is the inner covering, and this is the intermembrane space. And there are certain kind of carrier molecule which are present on the membrane through which electron are transferred. Right, and protons are built up inside the thylakoid. Okay, so this is the stroma, this is the inner thylakoid membrane, and very when it is collecting in the inside of the thylakoid space, it will then further diffuse utilizing F0 F1 particle and then produce ATP. Here our enzyme ATP synthase is used up for this particular process. Okay, so already we have seen this picture just a few PPT before, where in PS2 splitting of water occur and oxygen is evolved, and these are the proton molecules, and electron are passed from this blue dotted arrow showing the movement of electrons. Right, and this red one showing these arrow, broken arrow, showing the collection of or movement of proton. Okay, in thylakoid lumen, and finally it is collected in the lumen side, and then utilizing ATP synthase it pass F0 F1 particle and further used up by the ADP in the presence of inorganic phosphate and produce ATP. So what are those key components involved in this proton gradient? First is the development of gradient, that is known as proton gradient, means accumulation of proton inside the thylakoid membrane. Just now we have seen the picture inside the thylakoid membrane. Second is, first is proton gradient, another one is enzyme ATP synthase. Okay, and this enzyme is essential for ATP production. F0 subunit, it actually transport proton across the membrane. F1 subunit catalyze ATP synthase where when activated by the proton flow. Okay, so this is the F0 and this one is the F1 particle. This F0 is actually catalyze the movement of proton, while it is involved in the transportation of these protons from inner thylakoid membrane to the outer side, and this F1 particle catalyze the ATP synthase when activated by the proton flow. Okay, and the proton pump, this proton pump, so first is proton gradient, second is ATP synthase, this ATP synthase, and then these two subunit F0 F1 subunit, and third one is proton pump, which facilitate the movement of proton across the membrane. Okay, so in the light reaction, chlorophyll absorb light. We know this, chlorophyll absorb light, which actually initiate electron flow and proton release from the water molecule. Electron transport occur. Excited electron move from one carrier molecule to another carrier molecule utilizing electron transport chain, right? And the proton accumulation occur inside the thylakoid space, and these protons are pumped into the thylakoid space through proton gradient, and this movement of pumping establish a proton gradient, and during this process NADPH is formed. This proton contribute to the reduction of NADP to NADPH in PS1, and this gradient utilization, proton return to the stroma through ATP synthase F0 subunit, and it release energy, right? So NADPH formation occur and ATP formation occur, in which energy release power the F1 subunit to synthesize ATP from ADP and phosphorate. Now, where the ATP and NADP used up, this ATP and NADP is produced during photosynthesis, right? In osmotic pathway and in light reaction. But where these energy currency or energy molecules are used up, we know this, that ATP and NADPH produced in the light reaction in photosynthesis, and these are actually used in biosynthetic phase to produce sugar. What is the main function of photosynthesis? Production of food, production of starch, right? So this energy ATP and NADPH is further used up in the production of sugar. Okay, and what is this phase is known as Calvin cycle. This Calvin cycle does not require direct light, but it depends upon the ATP and NADPH, which is produced during light reaction. Okay, food or sugar is produced during Calvin cycle. We have discussed this previously also. But for the production of sugar, this Calvin cycle require ATP, NADPH, carbon dioxide and water. Clear? So, ATP and NADPH drive the conversion of carbon dioxide into the sugar. Okay, and this is produced during light reaction and used up in the dark reaction for the production of sugar. Okay, so the initial product which is formed, it can vary. In C3 plant there are two type of plants, C3 plant and C4 plant. So C3 plant, the first product is 3-phosphoglyceric acid, PGA, 3-PGA, 3-phosphoglyceric acid, right? While in C4 plant, first product is oxaloacetic acid or OAA. This is oxaloacetic acid. Okay, this is C3 plant and this one is C4 plant. Clear? So, first product is here, first product is PGA, 3-PGA phosphoglyceric acid, while in C4 first product is oxaloacetic acid. This distinction in carbon fixation pathway is based on the first stable product formed after carbon dioxide assimilation. Okay, in C4 plant there are two type of cells are present in the C4 plant. Some are mesophyll cells, some are bundle sheaths. we can classify it. While here only one type of cells are present, that is mesophyll cells. Okay. So now let's discuss the discovery of ribulose bisphosphate. Ribulose bisphosphate. So scientists initially sought to understand which molecule, after accepting carbon dioxide in the dark reaction, would yield 3-carbon product like PGA. Which molecule, after accepting carbon dioxide in dark reaction, would yield a 3-carbon product like PGA. So surprisingly, studies reveal that the primary acceptor molecule is a 5-carbon ketose sugar known as ribulose bisphosphate, RUBP. So this is the first acceptor or primary acceptor which actually accept this carbon dioxide. Okay, so scientists had initially hypothesized that a 2-carbon compound as a primary acceptor, leading to the years of research before discovering ribulose bisphosphate role. Okay, now let's discuss this dark cycle or Kelvin cycle. This Kelvin cycle was discovered by Kelvin and his colleague, and this he operates cyclic. This is a cyclic process. Kelvin cycle is a cyclic process which actually regenerate ribulose bisphosphate, and this is the primary acceptor. Okay, so this Kelvin cycle occur in all photosynthetic plant, whether it is C3 plant or C4 plant. Okay, so this is the light reaction which is utilized light energy of the sunlight, and splitting of water occur, releasing of oxygen, electron and proton occur. Okay, electron and proton further utilized uh, by electron transport chain and proton gradient for the production of ATP or NADPH, and this product of light reaction is further used up by the Kelvin cycle, in the Kelvin cycle, to fix this carbon dioxide. And what is the primary acceptor? That is ribulose bisphosphate. Okay, so this Kelvin cycle completes in 3 steps. First step is carboxylation, then reduction, and third is regeneration. And this, we have discussed this, ribulose bisphosphate is the primary acceptor of carbon dioxide, right? So first step is carboxylation or carbon fixation. What is the first step? Carbon fixation. So this carbon dioxide from the atmosphere is fixed into organic intermediate. Okay, and for this fixation of this carbon dioxide, some enzyme is involved, and then what is the name of enzyme? Rubisco enzyme. Rubisco enzyme. Ribulose bisphosphate carboxylase oxygenase. Okay, so Rubisco enzyme is involved for this carbon fixation process. So this carbon combined with ribulose bisphosphate, this is the primary acceptor, as I told you, right? So this carbon dioxide combined with this ribulose bisphosphate to form a two molecule of 3-PGA, phosphoglyceraldehyde. 2 molecule of 3-PGA, right? After that, second step. What is the second step? Reduction. Means 2 ATP molecules are used to phosphorylate this 3-PGA. 2 molecule of ATP and 2 molecule of ATP is used to phosphorylation, and 2 molecule of NADPH are used to reduce phosphorylated PGA into glyceraldehyde 3-phosphate. Okay, so here 2 molecule of ATP molecule, 2 ATP molecule and 2 NADPH is used up in this reduction process. Okay, so after 6 cycle, 1 glucose molecule is formed. Okay, as you know, this ribulose bisphosphate is a 5-carbon molecule, and this one is the sixth carbon. So after completing 6 cycle, one glucose molecule, which is a 6 carbon sugar, is formed. Okay, so for this 5 G3P molecule are used to regenerate ribulose bisphosphate. Clear? Now, third step, which is known as regeneration. Okay, so after reduction, it will convert into triose phosphate. Okay, so after this third step, regeneration means regeneration of what? This ribulose bisphosphate, which is generated to accept a new carbon dioxide. It will again regenerate to accept. This will regenerate after regeneration step to accept this new carbon dioxide through carboxylation. Okay, so this is the third step. For first step, carboxylation, followed by reduction, in which ATP and NADPH is used up, and then it will, after reduction, it will just leave that carbon and fix it into sucrose, and then regeneration of this ribulose bisphosphate occur. Okay, why we require this ribulose bisphosphate? To accept a new carbon, and in this process one ATP molecule is used up. 1 ATP. So here 2 molecule, here 1. So total 3 ATP molecule is used up in this process. Okay, and 2 molecule of NADPH. So per carbon dioxide molecule, 3 ATP, 2 over here, 1 over here, right? 2 NADPH is used up for the production of 1 glucose molecule in 6 cycles. Okay, clear? So 6 carbon dioxide, total number of ATP 18 ATP, total number of NADPH 12 NADPH, right? So 6 carbon dioxide is utilized for the production of 1 glucose molecule. Okay, so 6 carbon dioxide molecule, 18 ATP and 12 NADPH. Okay, in 6 cycles, so 2 and 1 3, so into 6, 18 into 6, 12. So why 6? Because in 6 cycle, one carbon dioxide per cycle, and we need 6 molecule for the generation of glucose. Glucose is a 6 carbon molecule, right? So this is how we can conclude to fix for the generation of 1 glucose molecule, we need 6 carbon dioxide molecule, 18 ATP molecule and 12 NADPH molecule. Clear? Now, let's discuss C4 plants and C3 plants. So, how this C4 plant is differentiated from C3 plant? First of all, if we compare the leaf anatomy of both the plants, the C4 plant have a specific type or specialized type of anatomy known as cranch anatomy. Okay, this cranch anatomy can withstand high temperature and intense light conditions. And it doesn't have, it lack photorespiration, leading to higher biomass productivity. Higher biomass productivity. Okay, so what is this cranch anatomy? Means large bundle sheath cells are surrounded by vascular bundles. This is the C4 type leaf. This is the C4. This is the cuticle, upper layer, followed by upper epidermis, right? These are the mesophyll cells, intact cells, and this inner one is the bundle sheath. Okay, so large bundle sheaths are present over here. While if you compare it with the C3 type of plant, fine layer cuticle followed by upper epidermis, then palisade mesophyll cells. Okay, palisade followed by spongy mesophyll cell, palisade mesophyll cell, then spongy mesophyll cells. Okay, and then vascular bundles and some intracellular spaces are there. There is no bundle sheath present in the C3 plant, while bundle sheath is present in the C4 type of plant. So large bundle sheath cells surrounded by vascular bundle sheath cell have numerous chloroplast, thick walled, and there is no intercellular space. There is no intercellular space. Okay, while there are somehow here it is not cleared, but some a kind of space are present in the spongy mesophyll cells in C3 plant. Okay, so there is no intercellular space in bundle sheath. Now, H and slack pathway. This is a pathway is used up for the carbon dioxide fixation. Okay, in C4 plant. In C4 plant. So, carbon dioxide, this atmospheric carbon dioxide is first fixed in mesophyll cells. This is known as mesophyll cells. Okay, it comes and accepted by a molecule in the mesophyll cells. Okay, and what is the primary carbon dioxide acceptor over here? It is a 3-carbon molecule known as phosphoenol pyruvate, PEP, phosphoenol pyruvate, which actually accept carbon dioxide in mesophyll cells. Here, an enzyme known as PEP carboxylase or PEPase catalyze the reaction between carbon dioxide and phosphoenol pyruvate and form a 4-carbon molecule known as oxaloacetic acid or OAA. So this is the 3-carbon molecule, and then after accepting 1 carbon dioxide, it will convert into C4 acid. And what is that molecule? Oxaloacetic acid, that is 4-carbon molecule. Okay, and after that, this 4-carbon molecule or oxaloacetic acid is get convert into another 4-carbon molecule known as malic acid or aspartic acid within this mesophyll cell. All this conversion occur in mesophyll cell only. Okay, after that, this C4 acid means malic acid or aspartic acid further transported into the bundle sheath cell. These are then transported into the bundle sheath cell. Okay, and in bundle sheath cell, the C4 acid, this C4 acid is broken down into 3-carbon acid, and the oxygen which is accepted by the PEP initially is released and here as a carbon dioxide molecule through decarboxylation. This is known as decarboxylation. So, malic acid or aspartic acid then transferred from mesophyll cell to bundle sheath cell where decarboxylation process occur, and through decarboxylation 1 molecule of carbon dioxide is released up, which is further enter into the Calvin cycle, and then again the formation of C3 acid occur. This is further transported back to the mesophyll cell. So this is how this phosphoenol pyruvate get generated again. Okay, so the released carbon dioxide enter into the Calvin cycle in the bundle sheath cell where it is used to synthesize sugar. Okay, so Calvin cycle occur in the bundle sheath cell in C4 plants. Okay, now the 3-carbon molecule produced during the breakdown of C4 acid is transported back to the C3 mesophyll cell, as I said just now, and in mesophyll cell this 3 carbon molecule is converted back to the PEP, which complete the cycle. So this is how fixation of carbon dioxide or carbon fixation occur in C4 plant. Clear? Now enzymatic presence. The bundle sheath cell in C4 plant, it is rich in rubisco, ribulose bisphosphate, carboxylase oxygenase enzyme, and it doesn't have PEPase. This PEPase is present in the mesophyll cells only. Clear? And in mesophyll cells doesn't have this rubisco, but it is having PEPase. So what is the summary? In C3 plant, Calvin cycle occur in all mesophyll cells, while in C4 plant, Calvin cycle occur only in bundle sheath cell, enhancing its efficiency in hot and dry environment. Clear? Now, what is photorespiration? Photorespiration is a process that affects C3 as well as C4 plant differently, as this photorespiration involve enzyme rubisco, which catalyze the first step of Calvin cycle, means combining this ribulose bisphosphate with carbon dioxide and to form 3-PGA. Right? So it actually catalyzes the first step, and this rubisco can bind both with carbon dioxide, that's why carboxylation, and with oxygen, means oxygenation, that's why it is known as rubisco. It is having both kind of activity, carboxylation and oxygenation, right? So rubisco can bind both carbon dioxide and oxygen, and its binding is competitive, so it determined by the relative concentration of carbon dioxide and oxygen. If carbon dioxide is more present in optimum amount in the environment as compared to oxygen, it will able to bind with the carbon dioxide. If carbon dioxide is not present, it is very easily bind with the oxygen. So it can bind both type of gases, carbon dioxide and oxygen, and its binding is competitive. So in C3 plant, some oxygen can bind to the rubisco and reducing carbon dioxide fixation in C3 plant. In C3 plant only mesophyll cells are there. Okay, so it can bind with the carbon dioxide, oxygen. So the ability of rubisco to bind with the carbon dioxide is less. So while so instead of forming, so in this type of condition, instead of forming 2 molecule of 3-PGA, which is normally happen during Calvin cycle, this RUBP, rubisco bisphosphate, bind with the oxygen to form 1 molecule of phosphoglyceraldehyde and 1 molecule of phosphoglycerate glycolate. Okay, so rather convert it or binding it, or we can say forming 2 molecule of PGA, it will bind with the oxygen if oxygen is present more in the environment, and form 1 molecule of phosphoglyceraldehyde and 1 molecule of phosphoglycolate in the case of C3 plant. And this process is known as photorespiration. Photorespiration, which does not synthesize sugar, ATP or NADPH, but result in carbon dioxide release and ATP utilization. Okay, this is known as photorespiration. While in C4 plant avoid photorespiration. How? By increasing carbon dioxide concentration at this enzymatic site. So obviously enzyme bind with the carbon dioxide. Okay, and C4 acid are broken down into the bundle sheath cell. As the C4 plant acid are broken down in the bundle sheath cell, just now we have seen the diagram. Okay, and in bundle sheath cell these enzymes are present, so it function primarily as a carboxylation or carboxylase. So this mechanism enhance productivity of the plant and yield of that particular plant, and provide greater tolerance to high temperature. So because of the different kind of anatomy, Crange anatomy, which is present in the C4 plant, it will avoid photorespiration and it will fix all the carbon dioxide present in the environment. Okay. Now let's discuss the factors which affect photosynthesis. There are so many factors comes under internal factor category or external factors category. Internal factors like leaf number, leaf size, age of leaf, orientation, type of cells like mesophyll cells or bundle sheath cells, stomata, internal carbon dioxide concentration, chlorophyll amount. These are comes under internal factor category. External factors like sunlight availability, temperature, carbon dioxide concentration, water, these are comes under external factor category. Okay, these all factors affect photosynthetic rate. One usually limit it. Okay, so law of limiting factor. Let's discuss law of limiting factor. If the process is affected by multiple factor, means sunlight, intensity, water content, and so on. So, though so many factors are there, that time the photosynthetic rate is determined by the factor nearest to its minimal value. Okay, so that time that factor act as a limiting factor. Like light, quality, intensity, and duration of the light are the key factors. If light is present in low intensity rate, then it shows linear increase in carbon dioxide fixation. Okay, but after reaching a optimum point, there is no increase, right? High light rate plateau as other factors are limited at that time. And saturation at 10% full sunlight. Excess light can degrade chlorophyll. Only 10% full light is used up by the plant for photosynthesis purpose. Carbon dioxide concentration, it is also act as a major limiting factor. Normally in atmosphere carbon dioxide concentration is 0.03 to 0.04%. If carbon dioxide is present in higher amount, it will increase fixation up to 0.05%. Now let's compare C3 with C4 plants. If light is present in a low amount, intensity is low, then neither respond to high carbon dioxide. If high light is present, in that case, in C3 and C4 plant, both increase the rate, photosynthetic rate. C4 saturate at 360 microliter per liter, while C3 450 microliter per liter. So in greenhouse, this light act as a limiting factor. So it is used in the greenhouse to boost productivity of the crop plant, like in tomatoes. Third is temperature. Dark reaction are temperature controlled. In C4 plant, high temperature is optimum, while in C3 plant, low temperature is optimum. Right? So it depends upon the habitat. Habitat type is tropical or template. Next is the water content. Water actually indirectly affect the plant health. Sometimes water is present in the excess amount, present in excess or extremely low condition. That time it acts as a limiting factor. If water stress is there, then stomata automatically closed up. So it will reduce carbon dioxide availability also. Okay, because from the stomata only gases are passed inside and outside environment. So under water stress condition, stomata will get closed and it will reduce carbon dioxide availability for its fixation. Water stress also cause leaf wilting, reduce surface area and its activity. So this is all about the photosynthesis topic. Now in two parts, part 1 and part 2, we have completed this particular topic, photosynthesis in higher plant. If you will find any type of difficulty in this particular chapter, have any doubt, please let me know. We will discuss it later on. Till then, take care, bye bye, and keep learning. Namaskar.